See my laser pointer?
Beautiful. Take it away.
Okay, sorry. Oh, my laser pointer is a little bit weird on my screen. That's all right. Okay. Good morning, everyone. I am Rebecca, and I will be presenting to you my confirmation of candidature for my PhD project entitled Radiation Therapy for Tumour Microenvironment Modulation to facilitate immunotherapy treatment of mesothelioma. So one of the hallmarks of cancer that distinguishes it from normal cells is its ability to evade the immune system. Our immune system is constantly checking for and destroying any irregular cells that it doesn't recognize itself, and this includes any of our own mutated cells. In order for a tumour to proliferate and survive, it is able to trick the immune system into recognizing it itself. So one cancer treatment is immunotherapy, and one type is anti-CTLA-4. So antigen-presenting cells present cytotoxic T cell lymphocyte-associated protein 4 to activated T cells, and then this downregulates an immune response. The anti-CTLA-4 immunotherapy blocks this binding, and the immune system mounts an immune response. Another immunotherapy is anti-PD-1. So tumor cells present programmed death ligand 1 to programmed death protein 1 on activated T cells, and then there is no immune response. There are two immunotherapies which target both sides of this binding, but for my project we'll be using anti-PD-1, which targets the T cell. So not all patients respond well to immunotherapy. There is this common trend seen in lots of different cancers treated with lots of different types of immunotherapies, where some patients respond really well while others do not. We're trying to figure out why this is. So immunotherapy is relatively new. It was only created in the 90s, and there's still so many unknowns. And one of the key factors in response is due to the differences in the tumour microenvironment. And we believe that radiation can be used to increase the effectiveness of immunotherapy. So radiation therapy is great for localized treatment, but it can be difficult to treat any dispersed microscopic or metastasized diseases. So when you irradiate a tumour, it releases tumour-specific proteins, which the immune system can then pick up and recognize, and can be used to detect and destroy other tumour cells. So this can be seen in the abscopal effect, and I'm sure we're all aware of this remarkable case where a patient was receiving palliative care, so palliative radiation to a few painful locations, and it resulted in a whole body immune response. And while the abscopal effect is impressive, it doesn't always occur and is incredibly unpredictable. So we believe that combining RT and immunotherapy, we're able to increase the specificity of immunotherapy. So why is this? In response to low non-cytotoxic doses of radiation, tumors create novel peptides for antigen presenting, and these are increased in expression. They are presented on major histocompatibility complex class 1s, which are also increased due to the radiation exposure. So the MHC1s present the increased peptides to CD8 T cells or cytotoxic T cells. They are also increased in number due to the radiation. They're assisted by CD4 or helper T cells that release cytokines to regulate other immune cells, and tumor infiltrating lymphocytes are also increased. And this is an umbrella term for all different sorts of white blood cells, or immune cells such as natural killer cells, dendritic cells, macrophages, all of those. And the radiation also causes an increased expression of tumor necrosis factors. And radiation also has a larger effect on the tumor vasculature. So normal tumor vasculature is organized and well structured, and all parts of the organ or tissue receive blood and nutrients. But tumor vasculature growth is very fast and unregulated and results in dead ends and malformed blood vessels. This results in poor blood and nutrients delivery to parts of the tumor. But low doses cause the activation of vascular endothelial growth factor, which stimulates the growth of normal vasculature, and this reoxygenates the tumor. So here is a good diagram explaining how both high and low doses of radiation impact the tumor vasculature and the immune system and how they interact. It's a little bit complicated and messy with a lot of acronyms that no one's quite sure what they mean. So to summarize, radiation can cause vascular remodeling of the tumor, which increases blood and oxygen supply. This then increases the perfusion, so the drug can be delivered into the tumour more effectively. RT activates and upregulates the immune system, allowing for more reactive immune cells. And it also helps the immunotherapy be more specific due to the release of tumour-specific proteins. And one type of cancer we think will benefit from immuno-RT is mesothelioma. So mesothelioma is a cancer of the mesothelium, which is the protective lining that covers all our organs and cavities. And it is caused by asbestos exposure with a latency period of, on average, 40 years. So asbestos was widely mined and used in Australia, and due to this, there are around 800 new cases diagnosed each year. This number is expected to increase as asbestos-riddled infrastructures are renovated and removed. So mesothelioma has the lowest 5-year survival rate of any cancer in Australia, with patients surviving on average 11 months after diagnosis. The treatment of surgery, chemotherapy, radiation therapy, and immunotherapy do not have that much success, and no one treatment option is ideal. They each have their own risks and side effects. To overcome these limitations, we want to combine treatments to have a synergistic effect. This has been seen in preclinical research. So here, murine models of AB1, AB12 mesothelioma found that the combination of anti-CTLA-4 plus immunotherapy slowed tumour growth as compared to just anti-CTLA-4 alone. And the secondary tumours, so tumours that didn't receive any radiation, had a growth delay, and some were even completely rejected, so the immune system destroyed them before they had a chance to actually grow into a tumour. And both the primary and secondary tumours had higher immune cell counts, which leads me to my project, which will be using radiation therapy to modulate the tumour microenvironment to help facilitate the immunotherapy treatment of mesothelioma in murine models. So first I did a literature review. So I looked into specifically non-invasive methods, how to image the tumour hypoxia, perfusion and vasculature of the tumour while it was still in the mouse, so in vivo, in the live mouse, no cutting out of the tumour. So there are different things to look at, including what they actually measure, such as blood or tissue oxygen or hemoglobin or a cell marker of hypoxia. The length of the procedure. It is very important for animal ethics that the mice be under anaesthetic for the least amount of time as possible. This is also important for the logistics of preclinical research, as we have a lot of mice that we have to treat. And if you have an imaging modality that takes a couple of hours, it can be difficult to treat the number of mice needed. The cost and availability of any contrast agents, and whether or not they have any side effects or can react with other things you're planning to inject in the mice. The depth and resolution of the imaging technique, and also how difficult and reproducible the results are of that imaging modality. So here's just a table summarizing a few of the main different hypoxia techniques. So as you can see, they all have different things which they use to determine tumour oxygenation. They all vary in length. Some involve non-reactive contrast agents, such as oxygen or carbogen breathing, whereas some have external agents such as radioactive FMC or fluorescent markers, which can have some negative reactions, and some use no external markers at all and simply rely on the contrast of the tumour itself. They all have differences in depth and resolution. So for my project, we're using valve-C female mice. These mice have a working immune system, and we'll be injecting them with AB1 murine mesothelioma tumour cells. The UWA School of Medicine is very experienced with these cells, and they have a well-defined and reproducible model. So for my first aim, I'm going to co-register different tumour hypoxia, perfusion and vasculature imaging modalities and correlate them to immunohistochemistry. So tumours will be imaged with these different types of modalities, which I'll get to shortly. They'll be co-registered and analysed and also compared to immunohistochemistry stains. So UWA has a new ultrasound-based imaging device, and we want to compare this technique to previous imaging techniques that have been used in order to establish a baseline. So the Vivo Laser X can image and quantify tumour oxygen saturation, vascularization and perfusion in a non-invasive way. It provides spatial information, and depending upon the technique, it can provide 3D or temporal images, and is non-invasive and very fast, which is is great for animal ethics. So to image hypoxia using the Vivo Laser X, we will be using the photoacoustic mode. So a laser is emitted from an attachment to the transducer. This laser light is then absorbed by the hemoglobin in the blood because it's coloured, it's a chromophore. This then causes the transient thermoelastic expansion of the hemoglobin, and those waves are then picked up by the ultrasound transducer. We're able to image both the deoxy and oxyhemoglobin due to 2 different wavelengths used. There is also tissue, we'll also be using tissue oxygenated level dependent MRI, which measures the relaxation rate of free oxygen molecules in the tissue, in the tumour. We're using FMESO-PET, in which the radioactive tracer accumulates in hypoxic cells. There are also commercially available fluorescent imaging markers, and one of them we'll be using is HypoxySense. So the CA9 antibody binds to the CA9 protein, which is a cell surface marker, and it is upregulated in hypoxic cells. And we'll be using the tissue stain pitomidazole, which again accumulates in the tumour, much like the FMESO does. So to image perfusion with the Vivo Laser X, we'll be using the contrast enhanced ultrasound mode. So non-targeted oxygen-filled microbubbles will be injected into the tail vein of the mice. They will then enter the capillaries, distribute throughout the tumour. And when we ultrasound this, we're able to distinguish the capillaries from the surrounding soft tissue. We'll be also using dynamic contrast enhanced MRI with a gadolinium contrast agent, which interacts with the water molecules of the blood, and this reaction is then detected by the MRI. The Vivo Laser X also has a power Doppler mode. So the movement of the blood causes a Doppler shift in the returning echoes, and this difference is picked up by the transducer, and an image of the blood vessels is made. And we'll be staining the tumours with anti-CD130 body. So CD31 is a protein of endothelial cells. So endothelial cells line all the blood vessels, and it can be seen here in brown. So to complete this aim, there will be 3 groups of mice. Each will receive the photoacoustic power Doppler and microbubbles, and one group will receive the MRIs, one will receive the PET, and the other will receive the fluoroscopy. But at the end, all tumours will be harvested and stained. Each group will be repeated 3 times, but there will be an additional mouse added for backup. So I hope to get 2 publications out of this: boxer-wise comparison of imaging modalities for tumour hypoxia and perfusion and vascularization, as well as the co-registration and correlation of imaging modalities and immunohistochemistry for tumour hypoxia and vasculature. So for aim 2, we want to determine the optimal fractionation schedule that primes the tumour microenvironment for immunotherapy. So tumour-bearing mice will be given 2 Gray and 6 Gray RT at different fractions. Imaging will be taken at different time points, and then they'll be compared to immunohistochemistry and flow cytometry of the tumour. So here is proposed treatment plans of the tumours. Tumours will vary in size not only due to the treatment they are receiving, but also due to the way that we inject the tumours. Mice will receive the prescribed dose, but the collimators used to deliver each dose will be adjusted for each treatment. So tumour-specific treatment plans ensures that we can cover the entire tumour, therefore the entire tumour microenvironment will be modulated. And there will be 8 groups being tested. So the photoacoustic and power Doppler will be done at each of these time points, but the microbubble injection ultrasound will only be done at these 2 time points due to expense. It is also quite difficult to inject into the tail vein of an anaesthetized mice. It is far harder than the manufacturers let on, so I'm also a little bit grateful that we only have to do it a limited number of times. So there will be 8 experimental groups, and each group will be repeated with 4 mice. And from aim 2, I hope to publish on how RT fractionation affects tumour volume, and also how changes in hypoxia, perfusion, and vascularization change with radiation dose and fractionation. Now, aim 3 is a big one. So we want to image and analyze tumour size in relation to time, dose, and fractionation of RT and immunotherapy. So tumour-bearing mice will be given the optimal fractionation schedule determined from aim 2, with different scheduling of anti-CTLA4 and anti-PD1. CT scans will be taken, and then they'll be compared to immunohistochemistry chemistry and flow cytometry. So here's the treatment schedule where we have 13 groups. This here will be determined by aim 2, so to be done. Not quite sure what fractionation we will need. So each group will be repeated with 10 mice, but then they will repeat it again for the 2 immunotherapy types. So I hope to publish how tumour size and immune markers change with the immuno-RT dose and fractionation. There is a lot more information that will be gathered from these experiments, specifically on immune cells, but this will be covered by another PhD student. So immune system is not my forte. And I'm hoping to publish my research, my literature review, on non-invasive preclinical imaging of hypoxia, and specifically how they compare to each other. So here is the budget taken from my research proposal. And here is my updated timetable I've readjusted for recent events. So what I have done so far off my timetable is my research proposal, where I wrote on the background of the immune system, the tumour microenvironment, and RT and immunotherapy, the aims of the project, the materials, the facility and the devices needed, as well as the methods of these devices, any skills and training that I have and will need to accomplish in order to achieve my aims, as well as the budget and the schedule. So we have to, one of the very first things, the most important things, is the animal ethics approval. Our ethics for aim 2 and 3 has been approved, where the ethics for aim 1 is under its final review. It can be quite a long process to write animal ethics. It can be around 6 months to write optimistically and then 3 months for it to go under review. It involves multiple meetings with multiple people from different facilities, a lot of coordination and several reviews for the committee. I wrote a large portion of aim 1 by myself, but I cannot underestimate the support given from my supervisors and others. It is a really quite difficult and is a major group effort. So I have had on my mouse training, my procedures. So I've been trained on subcutaneous injections for the tumour, intraperitoneal injections for the immunotherapy, tail vein injections for the microbubbles, as well as anaesthetic delivery on different types of anaesthetic machines and cervical dislocations. I've completed training on the small animal radiotherapy machine XRAD and its software. I used this during my master's, so I'm very familiar with it. It was relocated to the new Telethon Kids Institute building, so I had to do another building induction, a bioresources, and an XRAD refresher. So I'm classified as an autonomous user, so I can work by myself, but I can also supervise new users. And I've completed training on the LaserX and its software. So here's a photoacoustic image of a liver that I took. We're about to start defrosting and cultivating cells, which will take about 14 days to grow. Then we're going to inject them into a mouse and let them grow for another 14 days, so the tumour will be a substantial size. And then use those tumours to optimise some of these settings here. I was going to do that for the photoacoustic power Doppler and the microbubble, but given recent events, the labs have shut down and projects have been halted. So instead, working from home, I'll be working on some image analysis code. I'll be asking for some MRI, PET, immunohistochemistry, and ultrasound images from the different facilities and working with those. I will also be continuing on my literature review paper and also my introduction to my thesis, because that's a very long process and we've got time right now. And also our sponsors. So thank you. Any questions?
Well done. That was really interesting.
Thanks, Rebecca.
It's thunderous applause, Rebecca.
Yeah.
Okay, any questions for Rebecca? Rashid?
Yes. So can you tell me more about your radiation device? Because I have never seen it. I have no idea what it is like. What energy do you use?
It's 225 kV. So, oh, can you all see that still? Yep.
Yes.
Yeah, so the mouse lays here, will be lying at on its side, so the tumour's sort of up here and its head here and its tail's there. So doing that, yeah, 225 has got the copper or aluminium filter. It just rotates round 360, and the gantry can be moved up or down to the directions.
Yes, thank you. And are you using a specific dose rate?
Yes, I think it is set. What's the dose rate? I know that it's 13 milliamps. I'm not quite sure on the dose rate, but it's all set, and we can change it by the machine. But we'll be using the standard dose rate.
Right. Because are you going to test on different dose rates? Because in terms of radiobiology, the dose rate could affect. So, are you only using one dose rate, or are you also looking at using different dose rates?
So far we're only looking at one dose rate, but changing the dose rate would be very interesting. It would be interesting to see what's been done. I can see what has been done previously by Wes, who has just came before me, and see what dose rate he used and maybe change it depending on what results he got. But definitely changing the dose rate is something that could be looked into.
Right. Then you spoke about collimators. So what are your collimators like? Do you have leaves like MLC leaves that we have on Linux, or is it just?
It's just straight up, like, you know, circle. You have 10 millimeter, 25 millimeter, then you have circular, then you have a square as well. Right. And I do want to fiddle with those and see which gives us the best DVH. But I can't go in. Okay. So I need to see if the remote access is working, but they're just standard tubes, nothing fancy.
Right. Then Rebecca, so in reality, for patients, we treat them on a LINAC, don't we? So you're using a different dose rate and energy. How are you going to correlate your findings to what may be used in the future with like high energy LINACs?
Well, that is always the question with preclinical research, is, you know, how do you translate it? There have been lots of studies done on how well these devices translate to humans, and they've done it so that everything is scaled down. So the dose rate, the energy has all been scaled for the mice. So I wouldn't be so worried about that. But if you know, to translate better, we can use all this information that we get, and we're going to put it into a computational model done by Kelly. So you can put like mice information in and spit out some sort of human translation. So it's translating it as a bit of mix of what's been done previously. You know, we've been doing animal experiments for probably hundreds of years, and they've translated well, and then using new technology, new computers, new computational things that we have to actually refine these results better, so we can translate them more directly and just enhance them.
Okay, thank you very much.
Hi, Rebecca. We have a question here.
Okay.
It's about the KV machine.
Yes.
And me and Talat, we did similar work in KV machine in Canberra, and Talat is with me here. We have a question about the dosimetry. Is this machine traceable to ARPANZA, like in terms of calibration? Have you tested the dose, like 1 gray is really 1 gray? Or it has been tested before? Or?
Yes, yeah, it does go under regular QA, sort of. It does have a yearly QA that it goes under, and there's, I've sat on one of them, and they have, you know, the ball bearing and a cube that you use. It's got the lasers to figure out the ISO center, and it does have all the dosimetry that the human machines, maybe not as regularly, but they do have it done.
Yeah, as long as it's traceable to ARPANZA or other.
Yes, yes.
Yeah.
Martin does the QA, so you know it's good.
Yeah, so we also have a chamber that's been calibrated at ARPANZA.
Mm.
Yeah.
Right.
Mm-hmm.
Just to quickly, thank you for the presentation. It's very interesting what you are doing. Okay? Just quickly, do you have done any specific patient specific, like a mouse specific QA? Have you tried to see that what are your dose delivering is actually really delivered to the
One student did that with some 3D printed mouse models of gel, and they did some film calibrations with that. Yes, but we haven't really done anything where we like stick a diode in a mouse or something, or there's no specific sort of mouse phantoms.
Oh, okay.
But that's definitely something that I think should be done. Make a little phantom for the mouse.
When you try TLDs, it's much easier with the TLDs rather than just using like a film. That's too hard to do it. Like you can just run the same.
Yeah, definitely.
It's just good. Thank you.
Yeah, I think it'd be great to get a little mouse model, shove it, little diode in there and measure it.
Exactly.
Yes. Well, your project is full package. It's including imaging, treating, and biology. So it's very, very interesting. And it'll be interesting to see the MRI result in the future as well.
Mm.
And especially nowadays, I'm working with the ICPCM group for the MRI research. So if we think we need to include any things or any legislation, so we can introduce it to the ICPCM. Yeah.
Other questions? I have one. So when it comes to relating the imaging to the immunohistochemistry, how do you think you might manage to spatially align the images and the immunohistochemistry images?
That is something previous studies they've used they've used 6 to 8 mice, and they haven't been able to align like 1 or 2. So we doubled the amount of mice images that we're having. So if we can't align 1 or 2, it's still sort of statistically significant. But other people have used MATLAB programs. Another person used Photoshop, a few people. So they've kind of just done that. I want to do something a little bit better than that. I'm not too great with programming and everything. So what I think we're trying to do is we'll take a few slices of the immunohistochemistry from the center, and we will get the area, and we'll match the area to the largest area of the photoacoustic with the largest area of the immunohistochemistry. Also try and look for any maybe key sort of features like a little dent or a squiggle in the tumor, or any major vessels that we can see. But I think trying to match the area, the volume size, will be the best way. It is going to be a bit tricky, so we're going to be taking slices and images either side and just trying to find the best one, and then doing some sort of program to fully align them, which I will have to learn, and doing the computer vision unit will help. But I am expecting that maybe 1 or 2 might not be able to be fully aligned, because other people have had that problem. So just doubling the amount of images will help.
Basement.
Okay, Rebecca, how long would be the delay between your measurement for using image hypoxia and real treatment? I mean irradiation.
Pardon?
The interval between measurements for image hypoxia and actual treatment.
For the mice?
Yes.
There you go. No. Sorry, are you talking about here?
I mean the delay when you measure the hypoxia and the real treatment. So for example, if you measure hypoxia today and just going to treat it tomorrow, maybe the hypoxia is going to change. Is it immediately after measurements, or?
Other studies have found that sort of that changes to the hypoxia after sort of priming with the priming radiation, it can take usually takes about a day for things to start to change, because that takes how long it takes for everything to be released. So that's one of the things that we are going to have to figure out, because this schedule here is probably not fully set in stone. We might have to change some of the doses, spread some days out, determine, after we've done the first group, we're not maybe seeing much and we want to change things around. So I'm not quite sure exactly. We have to find that out through experiments.
That's great. So you are going to measure the hypoxia for different time intervals?
Yes, yeah, all these times here, and seeing how long those changes are sustained for, whether or not they only last a day or whether they're continued and all that stuff. So a lot of it's going to have to be, a lot of it's going to change, all the scheduling will change by what results we get from the first couple of tries.
Great, thanks.
Hi, Rebecca. It was a great presentation. It's a lot of work. I was wondering if you could go into just a little bit more detail on how you're going to compare the different techniques for AIM-1. I know you said it's going to be voxel by voxel comparison, but I'm not familiar with these or the output data or what that really means. So I was wondering how exactly you're going to compare them to say one's better than the other, or whether they're concordant, or what you're going to do.
I mean, I'm honestly not 100% sure myself. I'm not so great with computers and coding, but I think we'll have to compare each voxel and see sort of whether this one says, how hypoxic each little area of the tumor is. I think doing it maybe bit by region. So if we did, a little region here, a region here, a region here, and said, how each region compares, because the center is going to be more hypoxic than the external regions. So comparing regions, I think, would be a great way to compare the modalities. I think just seeing, because immunohistochemistry is classified as the gold standard for pretty much a lot of animal and this sort of thing. It's how they used to diagnose a lot of diseases in humans. So in order to compare the other modalities, I think comparing it to immunohistochemistry will be the key thing that will determine how well each of those modalities is, just because that is, the stains are what's considered the best and the most accurate. So I think, comparing region by region to each other and to the stains would probably be the best way. I don't know if does that help?
Yeah, no, it does. Yeah.
So Sarah Gulliford is at the University College London. She's one of our advisory panel members, and she's gone through your slides before today, because it's about 3 o'clock in the morning for her now, so she wasn't that keen to join. So she had a few questions, and one of them was, during this lockdown period where your experiments are on hold, are there any in silico simulations you can be looking at? So computational simulations of hypoxia that you might be able to explore?
I could certainly try. There aren't many simulations of it, because it's sort of, I honestly haven't looked into any of that sort of thing to try and do it, but I imagine there would be some models out there, but there wouldn't be any there would be very simple and complicated. They have models for like the immune system and equations and everything, but they're either very, very simple and don't have multiple factors, or they have like so many factors that it's almost impossible. So I mean, I'd certainly have a look around, but I don't know if there's many out there that will be quite as comprehensive, because our bodies are unbelievably complicated when you compare to just like the basics of radiation, particle enters, spurts out other things. when you try and include biology into it, it gets a little bit messy and it can be hard to have a simulation of it. But certainly take a look into it and see.
Brilliant. Other questions? Okay, well, thank you very much, Rebecca, and thanks.
Thank you, Rebecca. Well done.
Yep. Hey.
My first Zoom presentation, it feels a little weird.
So thank you very much for those people that joined us today. Really appreciate coming along as audience and for your questions and support. So we're actually now going to move to our normal Wednesday morning research update meeting, and you're welcome to stay and if you wish. But otherwise, yeah, thank you very much for joining us.